guys, this is Preeti from Hassan Institute of Medical Science. In this video, I'll be teaching you about the cardiac muscle histology, which is important question for the university exam, and you will be having your spotters of this. You have to write 2 points and identify the slide. Under the microscopic structure, you can see the cardiac muscle looks like this, and you have to write the 2 points. These are branched muscle fiber. Of like skeletal muscles are unbranched, smooth muscles are discrete in form, whereas the cardiac muscles are branched form. You can see intercalated disc between the two cardiac muscle fiber. This is an intercalated disc. The zigzag appearance of this thing is an intercalated disc. These are short, cylindrical compared to the skeletal muscle. Thus, muscles are short cylindrical structures than skeletal muscles, and you can see branching, whereas in skeletal muscles you see parallel cylindrical structures, whereas in cardiac muscle you can see the branching pattern. And intercalated disc is a unique feature of cardiac muscle, where this is present in between at the junction between the two cardiac muscle fiber. You can see intercalated disc, which works as a single functional unit and contracts at a time. And this branching cardiac muscle fiber, dark striations, you can see dark and light striations, but dark and light striations are not prominent in cardiac muscle, but prominent dark and light bands you can see in skeletal muscles. And intercalated is a single centrally placed nucleus, a single central placed nucleus, and sarcolemma. This cell membrane of this cardiac muscle is also known as a sarcolemma, and in skeletal muscle cell membrane also lemma, because sarcolemma, any muscle tissue cell membrane is known as a sarcolemma. And a few points which we have missed we'll see here. Short cylindrical fibers which are centrally placed oval nuclei, I have said, and the branching fibers you can see, intercalated disc is seen. So these cardiac muscle fibers are small and cylindrical compared to the skeletal muscle fiber. Nucleus is centrally placed. See, nucleus is centrally placed, oval in shape, and these are branches you can see here. And then intercalated disc is a unique feature of cardiac muscle. It is present at the junction between the two cardiac muscle fibers. This is the intercalated disc between the two cardiac muscle fibers. And zigzag appearance is made up of 3 types of cell junction, that is the desmosomes, gap junctions, and tight junctions. These are cellular communications, and it allows the electric impulse to pass from one cell to the another cell to make the cardiac muscle as a functional syncytium. Syncytium is together, it functions as a syncytium because everything will contract at a time because of this intercalated disc. And cardiac muscles is also striated, but striations are not prominent as such. Is in a low magnification we cannot see. If we increase the magnification, we can see these striations are prominent, but under low magnification, striations in skeletal muscles are more prominent than cardiac muscle. And cardiac muscle are involuntary, unlike in skeletal muscles. These are involuntary. We cannot control the function of the heart, and it is highly vascularized and innervated by autonomic nervous system. Heart, they are innervated by a parasympathetic nervous system, that is vagus nerve and all, and sympathetic and both parasympathetic will be there at a time. It the situation, according to the situation, both will be stimulated, which single thing will be stimulated, other will be inhibited. These are highly vascular. You know that the coronary artery supplies the heart. This is vascularized and highly innervated. This is a histological diagram which you have to draw for the exam. You have to draw the cylindrical muscle fiber with a centrally placed oval nucleus and the intercalated disc between the two cardiac muscle, and branching pattern you have to show. Single, and the membrane you have to label as sarcolemma. This is all about cardiac muscle histology. If you find this video helpful, please do like, share, and subscribe to my channel.